Good morning, students. I'm Jyoti, Head Department of Microbiology, going to mainly give you inputs about staining techniques in microbiology. So we know that microorganisms are organisms which cannot be visualized by our naked eye. That means bacteria are examples of microorganisms and they are generally colorless. So bacteria, they consist of a clear cytoplasmic or protoplasmic matter, and so they cannot be visualized in unstained conditions. In order to visualize them and to study their structure, shape and other structural characteristics, it becomes necessary to make them more easily visible under the microscope. So this means that the structures have to be contrasted from their environment so that they can be seen easily. So when we talk about mainly the microscope, which is the main instrument which is used for visualization of microorganisms, so we see that microscopy, light microscope is the commonly used microscope for observing the specimens in the microbiology laboratory. And the specimens can be prepared in two important forms. One is a wet mount technique and the other one is by staining the specimens. So what is a wet mount technique? Wet mount is a simplest type of preparation in which the specimen is placed on the slide in a drop of liquid. So when you take a broth culture, the broth culture contains the microorganism which is already present in a liquid form. So once the liquid is added to the slide with the help of a dropper, a cover slip is placed on the top and the specimen is generally ready for examination under the microscope. So, what are the main things what you can observe when you are using wet mount technique? Wet mounts are mainly used to observe bacterial motility. Bacterial motility indicates that microorganisms moving from one position to the another position. So if you want to observe pond water and the microbes present in the pond water, so we can use this method called as hanging drop method in order to observe the motility of the microorganisms. So how do you do this particular type of wet mount technique? So the procedure is we are going to suspend a small drop of the given suspension using a pasture pipette onto a slide. Then a cover slip is taken and it is gently pressed on the slide. Then the slide is placed on the microscopic stage and observed under the low power lens. And once it is observed under the low power lens to get a magnification of the organism, then it is mainly transferred to high power lens to increase the light and focus carefully. So bacteria should be magnified to sufficiently observe them. So the next important form is staining. Staining is imparting a color to the microorganisms so that a contrast is developed and the microorganism can be observed clearly under a bright colored background. So when we talk about stains, we have three important forms of stains based on their charges. So they based on the charges they carry: acidic stain, basic stain and neutral stain. So as the name suggests, acidic stain generally has an acidic charge on it. Examples are malachite green, grosin, Indian ink. Basic stains, they have a basic charge present on them. Examples are crystal violet, gentian violet, methylene blue. And when we talk about neutral stains, they contain both positive as well as negative charges, and examples are Giemsa stain, Leishman stain, Wright stain. So let's know the importance of acidic stains and basic stains. So we have already learnt about the examples of basic stains, and what do basic stains do? Basic stains or dyes, they bind to the negatively charged molecules such as nucleic acids, proteins and surface of bacterial cells or archibacterial cells. When we talk about acidic stains, they possess an acidic group like carboxylic group, phenolic hydroxyl groups, and they generally bind to positively charged cell structures. So let's mainly learn about staining methods. There are two important forms of staining methods, and in those two important forms, the first staining method what we are going to discuss is positive staining. Positive staining is a form of staining method where the actual cells are getting colored and they appear in a clear background. So we can categorize positive staining into simple staining and differential staining. Simple staining is mainly it provides color to the bacteria, and mainly it is used for observing the morphology of the bacteria. Example, methylene blue, carbol fuchsin, etc. But when we talk about differential staining differentiates between the bacteria and also the various structural components of the bacteria. Examples, gram stain, acid fast stain, and other important forms of special stains like endospore stain, capsule stain, etc. So let's learn about negative staining. So in case of negative staining, here what we observe is the cells remain uncolored and the background is colored to develop a contrast to aid the visualization of the image. And in case of negative staining, we use Indian ink and nigrosin. So when we talk about these two staining methods, that is positive staining and negative staining, so what are the various steps which are involved in these methods? The first step which is involved in staining procedure is smear preparation. So what do you mean by a smear? Smear is an even distribution of bacterial cells, or we call it as a thin film of bacterial cells on a slide for the purpose of viewing them under the microscope. So how do you prepare a smear? So what do we do is we take a small amount of the culture with the help of an inoculating loop and aseptically spread the sample over the surface of the slide. It is then left to air dry. This is mainly called as smear preparation, and the smear is now ready for the next step. So you can see this is a diagrammatic representation how the smear is prepared from liquid media and from solid media. So on the slide an area is marked, and within the marked area the small amount of the cultured is evenly distributed to form a thin film. So after the smear preparation, the next important step what we talk about is smear fixation. So when we talk about smear fixation, that is mainly fixing the smear onto the slide, it is by application of heat. The smear which is air dried, it is passed through the flame of Bunsen burner 2 to 3 times so that the smear gets fixed onto the slide, and then it is left to cool before staining. Now here we are talking about simple staining. In case of simple staining, it gives the morphology of the organism. A single stain is used, and examples of stains which can be used for simple staining, methylene blue, carbol fuchsin, and simple staining is of both positive as well as negative staining methods. So what is the procedure in case of simple staining? We are preparing a thin smear on a slide, heat fixing the smear by passing it 2 to 3 times gently over the flame of Bunsen burner. Then we are adding the stain onto the smear and allow it to stand for 1 minute. Wash the stained smear with water, air dry it, and observe it under the oil immersion lens. So you can see this is a diagrammatic representation. So we are first spreading the smear onto the slide with the help of the inoculating loop. Then air dry the smear, heat fix the smear. After heat fixation, then flood the smear with the requisite stain. Then you are going to wash away the stain and blot dry the specimen and observe it under oil immersion lens, that is under 100x lens. So these are few microscopic images. You can see microscopic images of bacteria under positive staining. So positive staining, the cells themselves get the stain here and the background will be clear. So you can see the microscopic background is clear and the cells have taken up the stain. So certain cells on the left hand picture are spherical in nature. They are mainly called as cocci, bacteria which are spherical in shape called as cocci, and the image on the right you can see that the bacteria are rod shaped and they are generally referred to as bacilli. So where we have already learnt about positive staining, now let's learn about negative staining. So negative staining procedure, it mainly helps to study the cell morphology. It is useful for only those bacteria which are difficult to stain, because there are very slender bacteria like spirochetes that are not detectable by staining methods, and they can only be viewed by negative staining method. So when we talk about the principle of negative staining, so in case of negative staining, acidic stains are used such as Indian ink or nigrosin. The acidic stain is negatively charged and it cannot penetrate the cell because there is also a negative charge on the surface of the bacteria. So like charges repel. So the stain will not be taken up by the bacterial cell. The cell remains unstained and then the stain is taken up by the background. So the unstained cells are differentiable against the dark background. So since heat fixation is not required here, the cells are not subjected to destaining effect of chemicals, and then their natural size, shape, arrangement can be observed by this method. So this is the main principle which is involved, where we see that bacterial cells which are very slender and which cannot be stained by normal staining method can be stained by negative staining, where the negative stain contains a negative charge on it and the bacterial cell repels this because it also has a negative charge, and the stain is taken up by the background. So what is the procedure for this kind of staining method, that is negative staining? So we can see here a loopful of undiluted Indian ink was placed at one end of the clean slide. Then a loopful of inoculum was transferred into the drop of the stain. Then by using a dirt-free slide with smooth edge over the suspension, it is spread uniformly along the edge. The suspension was spread to the end of the slide as to form a uniform smear, and then the slide is air dried and observed under oil immersion. The colorless bacteria are seen against a dark background. This is what we observe after negative staining procedure. So let me show you the observation of this. So this is the observation of your negative staining procedure. So in case of negative staining, the background takes up the color. The background here is mainly taken up with the Indian ink, which gives a bluish black in color, and what we observe is the bacterial cells are colorless, and those bacterial cells which are colorless can be clearly observed. Their size, their shape and arrangement can be observed. So these are the two important forms in case of simple staining, that is positive staining and negative staining. Similarly, apart from simple staining, we also are going to mainly learn about differential staining, and one important form of differential staining is gram staining. Gram staining is mainly used to differentiate the bacteria into gram positive and gram negative, and this staining method was given by a Danish physician Hans Christian Gram in the year 1883. So basically when we talk about this gram staining uses crystal violet, Lugol's iodine, acetone alcohol mixture and safranin. So here what we observe is the cell walls of gram positive and gram negative bacteria differ, and that is why there is a difference of the staining in case of gram positive and gram negative bacteria. So let me finally tell you that gram positive bacteria mainly appear purple in color and gram negative bacteria appear pink in color. So purple colored bacteria are always referred to as gram positive and pink colored bacteria are referred to as gram negative bacteria, and there's a basic principle which is involved. It is mainly because of the difference in the structure and the composition of their cell wall. And when we talk about the applications of gram staining is mainly used to appreciate the morphology and types of bacteria in clinical specimens. Thank you.